there are 280 participants registered from the ITS, while it is around 35 participants from the Saint La Salle University and nine participants from University Technology Mara. So in total, approximately, there are 302 to 324 participants from ITS, University of Saint La Salle, Philippines, and University Technology Mara. Among these participants, roughly, just for your information, that in Indonesia, the male Muslim participants will leave earlier, and it's around 100 and 125 participants that may leave earlier today. So basically, most of the participants will still continue to join this webinar, and because we also invite the participants from Saint La Salle University and also University Technology Mara. So if you don't mind, we would like to invite the representative from both university and also ITS to deliver 2-minute speech for these webinars. In this case, from the ITS, Dr. Indah Cisnawati will give the opening remarks from the ITS, while from the University of Saint La Salle, the Philippines, we invite Associate Professor Jessica Onate Pacalioga to give the opening remarks, maybe 2 until 3 minutes. And from the University of Technology Mara, Malaysia, we invite Dr. Nur Fatimah Muhammad Yunus for delivering the opening remarks. Hopefully, by inviting participants and delegations from the Philippines and Malaysia, represented by the Saint La Salle University and University Technology Mara, our collaboration will be expanded better. That's all from my side. Thanks to Dewi and Professor Inoue.
Thank you.
Thank you very much for your statement. Okay, so wait a moment, please. Wait a moment, please. それってもっと私だよね。あれ大丈夫ですよね。1分ぐらいになったからで、まして、じゃあ、割とあっさりと埼玉大学の話を、はい、ちょっとだけ、なんで今回されるんですか。でもあと5分。ちょっと授業早いんですけれども、今日のセミナーはインドネシアから生物環境系のお話の英語のセミナーになります。前から向いてることなのでね、これ今ズームで向こうからつないでもらっているんですけど、ちょっとよく言えば分かるんだけど、ズームに参加する人が300人を超えてるはずです。で、ここにいる学生以上に向こうの学生で、マレーシアとインドネシアの学生さんの顔を
masih celah enggak sih kalau bisa seperti itu ya. Terus. Nah, kita dipakai terus jadi
Bayangin aku ketemu Adika di gimana? Tunjukkan. Dia kenal aku gara-gara aku pakai ini.
Jessica.
Maksudnya apa?
Sekawan from Fatima from Malaysia and from Jessica for wrestling.
Excuse me, please off the mic without the lecture speaker. So I catch the sound audience speech talk. So it is complicated. Excuse me, please, all audience, stop your mic, please.
Not a participant, Prof.
Namanya kemana? Karena
Excuse me, Professor Toshio and Professor Naoya Inoue. Could you please give us thoughts to manage the session? Maybe to help this webinar with participants, please? Us, maybe. Thank you. The seminar very loudly with the non-mute participant. Could you please help us to be a co-host in this meeting so we can help you to mute the other participant? Thank you. I get over there.
Thank you for your comment. Can you hear me, everybody?
Very
Nice to meet
clear, very clear.
research staff of university and all students coming from Indonesia and Malaysia, Philippines. How are you? Fine. Everybody, it's okay.
Paling bawah loh. Oh, kok ngeceknya di PT pantes.
Okay.
Professor Inoue, sorry for interrupting.
Okay.
Professor Inoue, may I have your attention?
Okay. Okay. Attention, please. Attention, please. Thank you very much for your participation. Could you please yeah. Okay, thank you very much for your cooperation. So please cut off the microphone during the seminar. Thank you for your cooperation. Thank you so much for your participation today. My name is Inoue, calling from Saitama University. Saitama University is one of the national university of Japan. The Saitama University is very near to the downtown of Tokyo. Anyone who already have been come to Japan, maybe. So Tokyo is the capital of Japan, and Saitama is just kilometer only north of Tokyo downtown. So we have a national university, including chemistry faculty of chemistry and the liberal arts, and the faculty of science and the technology and the education. And so total number of students is more than 6,000. Anyway, so if you have a chance, student, could you please pay attention to Saitama University or Japan University? If you have a chance, please visit our university, pay attention to the Japanese university as a foreign student, international student. It's very nice opportunity. Can you hear me? All right? Anyone listen to me?
Yeah, clear.
Okay? Can you hear me? All right?
Professor
Okay.
Inoue,
Sorry?
Professor, if you don't mind, can you share the co-host for us so we can help to unmute the coming participants during the webinar so it will not distract our attentions.
Okay, thank you. Thank you your comment. All right. So today, fortunately, we could invite the Professor Dewi Hidayati-san.
Yeah.
Maybe pronunciation is very bad. Sorry. Could you please correct in your seminar, please, yourself? And she is coming from the Department of Biology, Institute Technology, Proof, November ITS, Surabaya city in Indonesia. So seminar title is this is also very difficult pronunciation for us. Toxicology and biology of fish for mitigating the impact of pollutants on aquatic ecosystem.
Tidak mengenal.
Thank you very
Jumpa
much.
pas Mehmet Gala ya. Aku ambil lo flash.
Okay. All right. Now, I wanna ask her to make a presentation in front of our students here. About 120 students, Japanese students are here, and also another several hundred students is connecting to the Zoom system at this moment. Okay, thank you very much for waiting for us. So I wanna ask Dewi-san to make a presentation just now. Could you please do it?
Sorry, Professor Noe, for the speech for the under university will be given before main seminar, or what about the speech from the delegates from the UITM, ITS, as well as from Sain Lasale? Pak Wahyu.
Ya apa? Oh ya, ini nama terus ITS.
Kalau ITS ya.
Sebentar ya.
Rita, please silent.
Thank you, Bu Dewi. Before you present your presentations, may I invite some representative from the Saint Lassalle University and also WTM and ITS to give the opening the remarks, maybe just 1-2 minutes each to welcome to all the participants and also the Saitama University. Maybe from the ITS, Bu Indah Trisnawati, please join us to deliver your remarks.
Okay, Pak Wahyu, Mr. Wahyu, thank you very much.
Ini apa lagi?
Is my voice is already here? Okay. Okay, thank you very much. Good afternoon, everyone. Konnichiwa, everyone. Okay, welcome to this international seminar. I'm Indah Trisnawati, representing from Faculty Science and Data Analytics of ITS. First, I would like to extend my gratitude to Saitama University in Japan for hosting this collaborative webinar as a part of HiSEP program. And I also want to express my appreciation to the staff, lecturer, and students from Saitama University, UITM Malaysia, and the LaSalle University in Philippines. Today we have the privilege hearing from a knowledgeable speaker from ITS who will share the important intersection of toxicology and fish biology within the framework of aquatic ecosystem management. I think this topic is important in relation to the challenge of our ecosystem face today. ITS is a national university in East Java, one of Indonesian provinces. Today, I would like to take a moment to highlight the Faculty of Science and Data Analytics as part of ITS. For your information, this faculty was established in 1965 under the initial name of Faculty of Certain Sciences and Natural Science. And until now, it has 6 departments: physics, mathematics, statistics, chemistry, biology, and actuarial science. I think with various fields in this Faculty of Science, there will be many opportunities for collaboration. Such as that. Excuse me, my mic is auto mute. Excuse me. I think with various fields in this Faculty of Science, there will be many opportunities for us for collaboration, such as student exchange for undergraduate research and postgraduate supervision. So for your information, the doctoral and master's program in all departments in this faculty have been internationally accredited by ASEAN. So that is my introduction representing ITS. I hope the collaboration will continue and be beneficial for all us. Thank you for your kind attention. I return the time to the MC. Thank you very much.
Thank you. Thank you, Bu Indah. Following this, could I invite Asok Prof. Jessica Onate Pacaliuga from Saint Lassalle University, Philippines, to give your remarks, 1 or 2 minutes. Thank you.
Hello, good afternoon. A pleasant afternoon to our partners at Institute Technology Sepulu Member and also our new collaborators from Saitama University of Japan and also the University of Technology Mara of Malaysia. Okay, again. Good afternoon, everyone. To our partners at Institute Technology Sepelu, Nepamber of Indonesia, also our new collaborators from Saitama University of Japan and the University of Technology Mara from Malaysia. I am Jessica Pacalyoga, the program head of biology of the College of Arts and Sciences, representing the University of St. LaSalle. It is indeed a wonderful opportunity for our universities to participate in this collaborative activity where our students and our colleagues can learn from a lecture presentation of selected topics. Last year, we had a very successful webinar jointly initiated by ITS and USLF. This year, we are happy to be invited again to listen to a very interesting topic on toxicology and fish biology, and also learn of ways to reduce the impact of pollution in aquatic environments. I am hopeful that this joint activity will inspire our students and our colleagues to pursue more research studies to be shared in collaborative webinars such as this. May this lead to a stronger partnership and more future engagements of our universities with each other. Good afternoon, everyone.
Good afternoon. Thank you very much, Professor Prof. Jessica Unati. And the last, let me invite Professor Asmida from the University Technology Mara, Malaysia.
Thank you, Bapak Wahyu. Good afternoon,
Nur
everyone.
Fatima.
I would like to extend the session to our head of department, Dr. Nur Fatima. Please welcome Dr. Fatima.
Okay, Assalamualaikum. Thank you, Dr. Asmida. Assalamualaikum warahmatullahi wabarakatuh, and very good day to everyone. It is truly an honor for me to be here today. I am Dr. Nur Fatima, currently head of School of Biology, representing Faculty of Applied Science, University Technology Mara, also known as UITM in Malaysia. And I would like to welcome all of you to this timely and important webinar, Toxicology and Biology of Fish for Mitigating the Impact of Pollutants on Aquatic Ecosystem. So first and foremost, I would like to express our heartfelt appreciation to the organizing committee team behind this initiative. And this webinar is the result of wonderful collaborations between Saitama University of Japan, the Faculty of Science and Data Analytics, ITS Indonesia, Institute Technology Sepelu November, and St. LaSalle University, Philippines, and our very own University Technology Mara, Malaysia. And thank you for your hard work and commitment to bring, I think we have like almost 200 students and participants to this event. So this team, the theme of today's webinar reflects a growing urgency in our regions and beyond. So the need to understand how pollutants impact the aquatic life and how we can apply the principle of fish biology and toxicology to mitigate these effects. So as we know, fish as bioindicators offer vital insight into the health of aquatic ecosystems, and they remind us that environmental stewardship is a shared responsibility. So at UITM, we take pride in building academic bridges across borders. We warmly welcome all partners, institutions, and all the students to explore collaborative opportunities with us. In particular, I represent UITM invite all students and lecturers to consider joining our outbound program for one semester at our faculty. So we believe that such exchange enrich not only academic growth, but also cultural understanding and long-term regional cooperation. So once again, thank you to all speakers, participants, and partner institutions. May today's webinar be a catalyst for a future research, dialogue, and collaboration. Inshallah, wishing you all a fruitful and engaging session. So back to you, Mr. Chairman. Thank you.
Thank you very much, Dr. Nur Fatima and Professor Asmida. Thank you very much for Professor Inoue to give us, to grant us the time to have remarks from our collaborator from the Philippines and Malaysia. And following this, please, I'll give back the presentation to Professor Dewi. Good Dewi, the time is yours.
Thank you, Pak Wahyu. Konnichiwa, mangandang hambon, selamat siang for all, for ladies and gentlemen. Firstly, I would like to extend my heartfelt gratitude to Professor Hasegawa, Prof. Naya Inoue, and all delegates and students from Saitama University, Prof. Asmida, Dr. Nur Fatimah Muhammad Yunus, and all delegates and students from University of Technology Mara, Malaysia, and Associate Professor Jessica Onete Pakaryoga, and all delegates and students from University of San Lasale, Philippines, and Dr. Indah Trisnawati, PhD, and all delegates and students from Institut Teknologi 10 November Indonesia, and all participants in this webinar. Good morning, I'm Dewi Hidayati from Department Biology of Faculty Science and Data Analytics, Institut Teknologi 10 November, or we can abbreviate ITS. Yeah, be presenting on the topic toxicology and biology of fish for mitigating the impact of pollutant on aquatic ecosystem. As we know, the pollutant is hello. Yeah. The pollutant is the main problem in all country. Effect pollutant can be measure, can be fish biology, yeah. Fish as a fish, the watery organism, yeah. Fish continually direct contact with any pollutant in the water body. They contact by the respiration system, the oral ingestion, thermal absorption, from the gill, heart, and brain. Yeah. The pollutant is a xenobiotic, the chemical substance that can adverse effect on aquatic ecosystem, as well acute effect, as well as the chronic effect. The chronic effect degrade habitat quality and lead to problems in fish, immune system, anatomy, physiology, reproduction, and development. So in the long term, it will be increasing the mortality and decreasing the biodiversity in the watering organism. I would like to maybe some participant is not from biology, so I will introduce you about the one of fish biology. As we know, the one characteristic of the aquatic organism, they respiration using a chill. The chill is different with the lung. The differences is the structure, but the function is the same. However, the structure of the structure of respiratory system in the chill have a characteristic of lamella. Lamella like a filament. Meanwhile, for the lung, for the terrestrial organism, the structure of the respiratory system is like the we can call a sac, like a sac. While in the aquatic organism, they have a lamella or filament structure. This filament structure is very tiny, only consists of one layer epithelial, and they continually receive the water, that contain the dissolved oxygen. The dissolved oxygen will be diffused to this lamella and will bring by the blood system to the all tissue and to all the cell in the body. Besides the dissolved oxygen in the pollutant water, in the pollutant aquatic, it's possibly bring the pollutant as well. So the chill, that continually receive the water, contact to the water, they have high risk to affect by the pollutant in the water, in the aquatic. Besides the fish biology, that can be as a biomarker or bioindicator for the aquatic pollution. The fish used as model in toxicology, like in the laboratory, because they have a small size, easy reproduction, short life cycle, and low maintenance cost ability to combine the genetic, cellular, and whole organism endpoints. By this advantage, there's a lot of research using fish model to evaluate the aquatic ecotoxicology, like a neural pathway, apoptosis increase, myocardial hypertrophy, activity, skeletal abnormality, embryonic, impaired inflammatory reaction. Fish have play crucial role in mitigating the impact of pollutant in aquatic system, because as a biologic in their biological, they can do biotransformation, they can do bioaccumulation. By this role, they can change in fish health and population, and we can measure of this change by using biomarker, and we can use as bioindicator. So the fish is biology. Fish plays a crucial role to research and mitigating the impact of pollutant in aquatic ecosystem. What is biotransformation? Biotransformation refers to the chemical modification of substance within living organism. The living organism, they have ability, to convert, the chemical that hazard to be not hazard, primarily through enzymatic reaction. This process is crucial for understanding how pollutants are metabolized by organisms, influencing their toxicity and persistence in environment. Biotransformation pathways are imperative to maintain the fitness of organisms. They facilitate chemical elimination from the body, thereby reducing bioaccumulation. This is the liver. The liver have main function for detoxification. Yeah. By their enzyme, they can decreasing the toxicity of the chemical and can eliminate from the body. This is the example of the enzyme that have main role for the detoxification. This is cytochrome P450 CEP enzyme. The CEP enzyme plays a prime role in biotransformers of broad range of exogenous and endogenous substrate. They respond to a wide variety of xenobiotic and therefore detect the presence of known and unknown pollutant relevant for organism. The highest activity of detoxification in the liver. Also found other organs like gonad, kidney, gills, and brain. Yeah. These genes highly expressed in endoplasmic reticulum or mitochondria, particularly hepatocyte, at least expressed in the brain. So we use this organ of fish, we can learn about the effect of pollutant and how the organism can respond to the pollutant. When the biotransformation is low or maybe lead the bioaccumulation. Bioaccumulation refers to the process by which an organism accumulates toxic substance faster than it can eliminate them. It possibly occurs when the water, the aquatic pollutant is very high. Yeah, so the fish, they can do biotransformation properly. So the toxic substance accumulation faster than elimination process. This leads to an increase in the concentration of this substance within the organism over time. This is several factors influencing bioaccumulation. Lipophilicity, substances that are fat soluble tend to accumulate more readily. Metabolic rate, organisms with slower metabolism may accumulate toxin more. Exposure duration, longer exposure period, increase accumulation risk. And this factor can lead to chronic toxicity and health in organism. May affect entire ecosystem, especially when the top predator accumulate in high toxic level. This is for the example of our research about the metal bioaccumulation in Oreochromis, tilapia fish. Yeah. The PP accumulation in the flesh of tilapia and its effect in the fish growth at different sublethal concentration. Our experiment by exposing tilapia fish for 13 days at different sublethal concentration showed that tilapia fish accumulated relatively high concentration at 30 at 10% LC50, but neither at the earlier incubation time nor other lower concentration. We also show that tilapia fish did not exhibit any significant growth inhibition at different sublethal concentration. We also have research of lucimat. Lucimat is abbreviated for the lumpur Sidoarjo. Lumpur is the name of mud volcano effluent. In Indonesia in 2006, there is we have occur the mud volcano that occur in the urban area, and this mud volcano emitted a huge volume mud in the end. This mud, this mud is channeled directly to the water. As we know, the mud from the mud volcano, it's not only consists of the mud, they also consist of maybe hydrocarbon material, as well as the heavy metals, because they emitted from about the 3 kilometers under the from the 3 kilometers from the earth, in the earth. This is when we study in situ, when fish body on metal in fish body that exposed in the mud volcano, mud flow, Lucy mud flow in the in situ on site in the river. We use this cage fish, and the fish is exposed in here. After 1 month, it's the control is about the aluminum is in the safe. However, when the condition of aluminum, that close to the effluent in P1 and P2, they have higher of aluminum. Maybe aluminum is not toxic. It's not toxic. However, where the pH is low, is very toxic. This is the example how we use the fish to identify the impact of the pollutant in water system in the aquatic. We use also the fish as biomarker. Biomarker is biochemical, cellular, physiological, or behavioral variation that can be measured in tissue or body fluid sample or at the level of whole organism that provide evidence of exposure to and or effect on one or more chemical pollutant. The metal toxicity, it's not just accumulate. Their accumulation in the body also have another effect, degradation effect, like the oxidative damage, hepatic lipid accumulation, oxidative damage, and vacuolating. I will show you my research about the effect of the heavy metal that make a vacuolating in the hepatocyte. Beside the ex situ, beside the mortality of the fish, we can also use a fish embryo, for a model. Fish embryo are used to study early development disorder. As we know, the fish, you do the external fertilization. So this development is very important to study. And the successful of the fisheries, it depends of the successful of the development of the embryo of fish. So this research is very important as well. Exposure is performed by fertilization to hatching a number of developmental Parameters include stage, summit formation, and heart rate, age development, pigmentation, tail lifting, hatching, behavior, and other will be observed. The fish embryo assays been used to evaluate effluent water from the oil production, pharmaceutical, and waste production. The other research, as we know, the microplastic also the one of problem in aquatic ecosystem in all country. Yeah. MPS impact on different organ on the fish body. They can change the feeding rate, growth rate, metabolic activity, and histopathological characteristic. I will show you some of research about the microplastic as well. We have research about the fish gill and scale as biomarker. Why? Because this gill, as I explained before, yeah, they continually contact to the water body because they continually take the dissolved oxygen, yeah. So it's very high, have high risk, yeah, to receive the pollutant, as well as the scale, because the scale in the surface of the body, yeah, they also contact to the pollutant continually. So it's very potentially as biomarker. Yeah. We use this fish Oreochromis to study the impact of pollutant to the structure and anatomy, yeah, histology, as well as the molecule and microanatomy of the scale. Yeah. This is a histological of the gill. This is a gill, yeah. This is a gill when we use the microscope, and this is a histological of gill. This is the gill. This is some our research regarding the impact of mud volcano with using the biomarker of fish gill chromatophore. Yeah. This is evaluation the effects of mud in aquatic life using chromatophore and microstructure, and using the microstructure and as well as the using biological study of fish. Yeah. Yeah. We use the in situ. It means the method is in situ experiment. The cage of tilapia fishes, Oreochromis mesamicus, were placed in downstream area that receiving the Sidoarjo mud flow effluent, and in the upstream as a control. This is the this is the figure, the control that very clear, yeah, and this is the location that received the mud volcano, very high of TSS, total suspended solid, is very high. Yeah. When we use the cage fish, the fish is placed in here. We not use the metal material. We use the other that not contain the metal material. Yeah. And after that, after about 30 days, yeah, the fish gill is exposed by the mud. They have a performa, yeah, this anatomy is a height of mud in this gill. And then we observe by histological analysis. Beside the in situ, we also study the mud sediment, yeah, that obtained from Sidoarjo mud pond area. We must definitely mention the coordinate. Yeah. Sorry, my battery is low. The watery mud was mixed with the fresh water in ratio 1:1 and used a treatment of media. Yeah. So the study, ecotoxicology study, we can do in situ as well as ex situ in laboratory. This is the other biomarker that we use is the scale. Yeah. This is the microscopic, use the light microscope, and this is use the electron microscope. Fish scale were removed using tweezer from the left side of the body between the dorsal fin and lateral line. The scale were cleaned with the sterile water. It's very simple to prepare. Yeah. And then we bring to the SEM analysis and chromatophoresis density using light microscope. This is the results for the chill alteration. After we expose the fish, yeah, the live fish in the polluted area, yeah, we observe histological characteristic, and we observe the histological alteration. This is a control. Yeah, as we know, the lamella or filament, yeah, filament is very regular, yeah, with only consists of one layer epithelial. Yeah. While, yeah, the histological structure of the chill that exposed to the polluted, you can see they have abnormalities. They high proliferation due to adaptation to protect the vascular system in here. So they because the pollutant, yeah, attack, yeah, the cell will the cell will proliferation, yeah, and make the normal is only one layer. That's many layer to be calling hyperplasia. Yeah, one layer epithelial to be multi-layer. This condition more safer in the 14 days, more safer. When each lamella high proliferation, yeah, so each lamella will fusion. It's so this fusion. As we know, I will show you the function of the chill. The function of chill depends of the structure. As we know, this is only one layer. When the lamella is fusion, so the dissolved oxygen is inhibited. So the diffusion of oxygen will be affected. So the respiratory system will be altered. Yeah, this condition lead the mortality chronic. Yeah, in the chronic, they lead the mortality of the fish. Yeah, And the other, we can see also in the side P, all P side, yeah, show the alteration of histological of chill rather than normal. And then this alteration can be scoring, and we call the alteration index. How to measure the alteration index? Yeah, we can use this table, this table. Yeah. For the example, yeah, this is we can use this score, yeah, epithelial lifting, lamella fusion, yeah, we can use this stage of the alteration from the reparable or reversible, and other is irreversible, yeah, like hypertrophy, hyperplasia of chloride cell. It's more safer, yeah. And after we scoring, we can make a score and classify the histological alteration status, the normal, irreparable, yeah, and slight damage, yeah. We can use define the status of the alteration, so the stakeholder can more easily to understand what the effect of the pollutant. Yeah, this is my other research using the XC2. Yeah, this XC2 using the mud volcano that we observe in laboratory. Yeah, this is that we observe in laboratory in XC2. Here. Yeah. Yeah, this is the XC2. Yeah. We try to remediate the mud volcano using a plant, yeah, and then we evaluate the toxic of water treatment. Yeah. We also using scoring of the liver histopathology, yeah, and then we scoring and we also observe the condition of histological. In the non-treatment water polluted by the mud flow, yeah, they show vacuolization of hepatocyte. Yeah. And other biomarker that we study is the chromatophore. Yeah. This is the structure of normal chromatophore. Yeah. The normal chromatophore, they have a lot of chromatophore, while the exposed fish that exposed by polluted water, the chromatophore is decreased significantly. Yeah, this is decreased. The chromatophore, this is a chromatophore. I will show you the structure of chromatophore. Yeah, this is a chromatophore. Yeah. Beside the in microscopic light observation, we also observe by using the electron microscope. Yeah, by the electron microscope, yeah, the chromatophore, the scale, they consist of the spherule. The spherule is the like for the attachment structure for the body of fish. Yeah, it's very important to attach the scale to the body of fish. However, in the polluted area, this spherule, yeah, the spherule is show the abnormality. So by this structure, it's possibly decrease the ability scale to attach to the fish body. This condition, yeah, will be dangerous if there is infection or predatory. Yeah, this is very infection affect the physiological of fish, especially toward the infection of bacteria. And after we calculate the number of chromatophore density, yeah, it showed that density of chromatophore in the control, it's mean they know polluted water, yeah, it's more higher than polluted water. Yeah. For the assessment, we also using the ecological quality ratio, so it's more easy to comparing to the biological quality elements. What is the ecological quality ratio? Is by divided the parameter value and reference value. However, it's depend on the reference value. For example, dissolved oxygen better in higher concentration. The reference value is 5.5 ppm. So the reference value, yeah, how to calculate the parameter value divided to the reference value, yeah, in this using this formula. However, for heavy metal, when the better in lower concentration, we use this formula. It refers reference value divided with parameter value. After we scoring, we can decide if the polluted area is poor or high or moderate. And this score can we use to evaluate the correlation between the biological parameter and the aquatic parameter, the water quality parameter. Yeah, this is a quality ratio that have score between 0 to 1. Yeah, when it's good quality when close to the 1. Yeah, and this quality ratio show relationship between chromatophore density and each quality ratio of total suspended solid and iron. Yeah, quality ratio correlation quality ratio and total suspended solid and iron falling within the level of 1 water quality and show higher chromatophore density. As we know, yeah, if the higher EQR, it means higher the water quality is better. The water quality is better, the TSS is better, or the TSS is low, the number chromatophore is high. While when the QR is low, yeah, like in the polluted water, it show the low quality ratio and affected the number of chromatophore. Besides the biomarker, the fish biology can be used as a bioindicator. A bioindicator is The definition of bioindicator is a detector that reveals complex condition resulting from combination of biotic and abiotic factor ranging from cellular level to ecosystem. Yeah. So that we learn previously also can use for bioindicator. This indicator quantify information, respond to change over time and space, and help characterize the state of an ecosystem by detecting early modification. This is my research about the use fish community, yeah, to evaluate the volcanic mud contamination in river ecosystem. Yeah, this is the area that I explained previously, that directly channeling the mud volcano every day, the huge volume every day, and this is the condition of gill fish in that we catch from the river. They a lot of mud, yeah, in the surface of the gillfish. Yeah, this is a condition of the control. While we study, we use the habitat quality index. Yeah, habitat quality. Why we use the habitat quality index? Because we hypothesize that hike of mud, yeah, will be change the substrate, especially the substrate will be have high percentage of the mud. Yeah, after we evaluation the quality index, yeah, the availability in stream cover, yeah, for the control is better than the polluted area. Yeah, because the one of characteristic of the stream cover, they must be height of material like a sand, yeah, like gravel. But when it's polluted by the high volume of the mud, it will be covering the stream cover, and the substrate, it's not stable. Yeah, so it will be decrease the value of habitat quality index. Yeah, dimension largest pool, number of river is relative okay, yeah, in the polluted area. However, for the dimension of the largest pool decrease, because the mud will be embedded in the river, so the dimension of the pool is increasing. After we evaluate the habitat quality index, we can determine the status is high, limited, and high, for the control is high, and the status of the quality index in Lucy receiving station is limited. After that, we learn about the component matrix and scoring method for index fish biotic integrity. Yeah, according to Moyle and Marchetti, we evaluate the percentage of native fish. Yeah, this is Moyle and Marchetti evaluation score. Yeah, we can it will be scoring low when the native species is less than 20%, while the native more than 80% will be scored more. Yeah, the number of native species present, number of age classes cyprinid, yeah, cyprinid, and total number of fish species, total fish abundance. Yeah, it's low or common or abundant. Yeah, percentage of carnivore. Yeah, when it's high, yeah, it will score better. Yeah, percentage of tolerant species. This is the highest or tolerant species is low score, while the tolerant species is low. Yeah, it's more better. Yeah. After we make a score, we can decide the status of aquatic communities: very good, good condition, fair, or poor. Yeah. After we learn about the composition and identification, we scoring, and we can found the scoring of quality fish biologic index. Yeah, the percentage of number of species. Yeah, as like a Moyle-Merchanti formula. Yeah, in the control is better. Yeah, after we scoring, yeah, the total score about 75 and 60. All this range is still good. However, by the scoring, yeah, the control is better than polluted water. It means the polluted water also the fish can adapt it to the polluted water that caused by the mud volcano. We also have research about microplastic characteristic found in gastrointestinal tract. Yeah. We also have used the fish to evaluation of water quality and survival rate tilapia using rice fish culture system in quarry land. Quarry land is the land, yeah, after the mining. After mining, there are the quarry land used to fish culture, rice fish culture. However, we must be we evaluate is the land is good for the tilapia or not. And we also have a collaboration research about the monitoring the metal concentration in the catch fish like a tuna. We also have research using fish to evaluate the blood profile and endoparasite in the culture floating marine cage. Culture using floating marine cage, because the floating marine cage, they have high of organic material potential to have a lot of high organic material. So we also study of the pollutant using blood profile. This is the other research using Tobara fish to indicator heavy metal pollution. This is the research that showed how important of fish as to BU indicator for environmental monitoring. Thank you for your attention. I will give the time for Professor Inouye. Thank you.
Okay, thank you very much for your presentation. I am honestly very interesting for your research field. Maybe focus on the fish and also very closely related to the, what can I say, social problem in the world, one of the social problem in the world. So solution is very, very important. Before that, the solution, maybe investigation is very important. So maybe your experiment or observation is very important for such kind of orientation to solve the social problem. Thank you very much. Any question? Still, we have a time. We have a time about 20 minutes or something. I want to ask some questions. First of all, according to the Japanese student in front of us, and then I want to ask to the international student, maybe connecting Zoom system. So if you have any questions, scientific question or not scientific question is available. So could you please say some questions to us? でいいんですかね? Any questions in front of a Japanese student? In Japanese, it's okay? No questions? Okay, think a question. We have a still time, okay? なんか長谷川先生あります？長澤先生もいいですか？ Okay, very simple question, very simple, stupid question maybe from me, from I. So, thank you very much for your presentation. So maybe you are focused on the experiment for concentrating to the fish. So, I have a question is river fish or sea fish, which is interested in for your experiment, for your investigation?
Maybe you can tell me. Maybe you can repeat again. I'm not clear voice.
I say, okay, I will ask again. So you make an experiment for the fish.
Yeah.
Maybe I suppose you are concentrating to make an experiment to the river fish, not the sea fish.
Oh, depends. Yeah. For this research, we use the Porong River is river, so we use the freshwater fish. So
Okay.
Depends of the location.
Okay, I can understand. And you mentioned some name of fish. So I noticed the fish name of fish is one of the fish is a milk fish.
Yeah.
We are not familiar milk fish in Japan.
Yeah.
That fish is a very, what can I say, popular in your country?
Yes, yes. We in Indonesia, in ITS area, we are in coastal area. The British fish, the milk fish is very popular in East Java, especially in Surabaya, yeah, because they large pond in East Java that culture this milkfish. The Porong River in Sidoarjo are also the center of milkfish. Yeah. Why we use the one ratio 1:1 because the mud flow itself is saline. Yeah. The mud flow is very saline. Yeah. So we use the fresh water to mix this media. Where this yeah, we use the ratio water, fresh water with the mud, because the mud itself is saline. Yeah. So after that, the media is brackish water. That's suitable for the milkfish.
Okay, I can understand. So, maybe Japanese people like very much fish food.
Yeah.
So
Also
maybe I
in
suppose
Surabaya, Professor, if the student come here, we will visit field trip to the mangrove area. That's a lot of ponds of milkfish as well.
Okay, thank you very much.
Yeah.
Any questions? No questions?
Professor, I think there's a question in the chat box from the University of St. LaSalle student. If I may read, this is a question from a BS Bio student with the name of Febi Gentile. Her question is, how might the regenerative capacity of gills and scales mask the visible impact of lead, or rather impact of lead, and does this affect the accuracy of these tissues as long-term biomarkers? Professor Dewi, were you able to get the question? It's in the chat box. Yes.
Yes.
Thank you.
Okay. In my research, we are not define lead as the pollutant, one of pollutant. In the mud volcano as a lot of heavy metal. So we cannot define it's caused by the only of lead or other pollutant. Yeah. So we use the other previous research reference, use the reference of the impact of the lead to the gill from the reference. Is my answer and get it?
Maybe if you can expand a little bit, but thank you for that answer.
Yes, this one. Yes, Prof. Or this one.
Okay, any questions? Asking too much? Okay, thank you very much. So honestly speaking, our Saitama University, maybe we are planning to visit Surabaya ICT University, ITC University, maybe in this September, maybe several students and some staff, maybe not so long, short-term visit to Surabaya, and then maybe we want to contact with you, and also we want to talk with your student. And also, if we have a chance to visit for Philippines or another country, so this is a very nice opportunity. So our scientific educational makes allow to us to visit, very short visit, unfortunately. So, but such kind of opportunity, looking for the opportunity is a very important, we think. This is one of the global education. So this is a very important action, maybe very common, what can I say, action, which every university require to the student or something like that. So one of this kind of international seminar is very interesting, actually interesting. So this opportunity could be increase. Of course, English, very good at English student, not good at English student. So some different. So, but a very good exercise, also very good exercise to study English through the scientific seminar. So if we can, we want to talk with the participant university. So we want to make another chance to make a English seminar from Japan, from Surabaya or Philippines. So maybe this kind of English, what can I say, series, English seminar series is very interesting. Maybe further discussion could be done with university staff among among this university. Okay, any comment, the staff of ICT University? Nothing special today?
The right hand one.
Hello,
So,
I'm good. Nasa breath na ko? Sabi ko yung hello. Si Akon.
Yeah. Yes, Prof. Inoue, we are very pleased to welcoming to the visit in ITS, and we have prepared. If we maybe we also can the student can learn how to measure the pathological using histopathological, yeah, how to evaluate the pollutant effect using histopathological study like this. Yeah, maybe we can the student from the Saitama can also learn how to evaluate using histological alteration of chill. And we also very exciting if the Saint Lazare University as well as the UITM Malaysia can make a program in the next program. We can make another webinar and other programs. We are hopefully it's not just now, but it can initiate to the other programs as well.
Yes, thank you so much, Dr. Dewi and also Professor Inui, for this wonderful opportunity for us to participate in this webinar. And yes, we look forward to more webinars in the future, even having our students share their studies as well. So thank you so much.
Me too. Thank you very much for your participation, and maybe this is very good opportunity. Maybe we are looking for the another opportunity in future. Thank you very much, everybody.
Yeah.
Thank you very much, students.
Thank you very much, Professor Dewa, Prof. Inui.
How can I say goodbye?
Yeah.
In Japanese, sayonara. Sayonara is a goodbye. It's okay, sayonara.
Bertemu lagi, selamat tinggal. Thank you, Dr. Fatima.
Okay, thank you very much.
Thank you, Prof. Jessica. Thank you.
Thank you. Thank you, everybody.
You can see you again in another program. Thank you.
Inshallah. Thank
Okay,
you.
bye-bye.
Bye-bye.